Okay, let me recording. Okay. And good morning, good afternoon, and good evening, everyone. And welcome to today's International Society of Biopharmaceutical Statistics ISBS webinar. My name is Haitao Pan. I'm honored to be your host for this session. It's a great pleasure to introduce our distinguished speaker, Dr. Jie Chen, today. Dr. Chen is the chairman and chief scientific officer of Taimei Intelligence Clinical Research, with over 29 years of experiences in biopharmaceutical R&D and life cycle management. His expertise include innovative study designs, multiple comparisons, Bayesian methods, real-world evidence studies, causal inference, and machine learning for real-world evidence generation. Dr. Chen has made significant contributions to the field, serving as co-chair of the ASA Biopharmaceutical Section Real-World Evidence Scientific Working Group and co-chair of the 13th International Conference on Multiple Comparison Procedures. He is also an editorial board member of the Journal of Biopharmaceutical Statistics and Contemporary Clinical Trials, and co-authored over 60 scientific papers and the book on medical product safety evaluation. Additionally, he is an elected fellow of the American Statistical Association. Dr. Chen is also one of the founders of the ISBS. Dr. Chen earned his medical degree from Shanghai First College Medicine, currently Fudan University, and a PhD in statistics from Temple University. Before we begin, just a few quick reminders. Please keep your microphone muted during the presentation. If you have questions, feel free to type them in the chat. We will address them during the Q&A session at the end. You may unmute yourself during the Q&A to ask a question directly. With that, let's get started. Please join me in welcoming Dr. Jie Chen. Dr. Chen, the floor is yours. Thank you.
Thank you so much. Hey, Tao, can you hear me? Okay, great.
Yes.
Thank you so much for the introduction, Hey, Tao, and thank you. Thanks to the ISBS International Society for Biopharmaceutical section to organize the webinar series and to give me the chance to share some of my work about the use of real-world data and real-world evidence in rare disease drug development. I would first to acknowledge my colleague at the ASA Biopharmaceutical Section RWD Scientific Working Group. You can see the name I listed here. We just have two papers accepted for publication by the clinical pharmacology and therapeutics. The first one is the challenge and the possible strategies to address them in rare disease drug development. The second will be the use of RWD and RWE in rare disease drug development. Today's talk will primarily based on these two publications. As you can imagine, we did a landscape assessment for the challenge and possible solution, including the use of RWD and RWE. And in my presentation, there will be no technical details on how to do to use RWD and RWE in drug development. And you can refer to some of the reference being provided through my talk. So today's talk will I will first give some of the introduction, and next will be some of the regulatory guidance, the guideline, initiative framework, etc. in rare disease drug development. And after that, I will discuss
Hi, Chen. Sorry,
Okay. Can you hear me now? Okay.
Yes, we can hear you. Thanks.
Okay, great. So and then we'll discuss the strategy to deal with the challenge, not necessarily about the use of RWE. And next I will discuss a little bit more in more detail in the use of RWD and RWE in rare disease drug development. And after that, I will briefly introduce the target learning-based framework. And you can, many of you may hear about the target learning framework already. And last year, I believe in June 2024, FDA published a demonstration project about the target learning framework being used in RWE generations. Before giving some of the conclusion and discussion and conclusion, I will present some of the case studies how show how RWD and RWE being used in rare disease drug development. So as you can imagine, each country or continent, they may have specific definition for rare disease. And the WHO definition is about 65 out of 100,000 people. That is something it's about the prevalence rate for the rare disease. In the US, the Orphan Drug Act, it is said as long as the condition affects less than 200,000 people in the US, that's equivalent to the prevalence rate 67 in 100,000 people in the US, will be considered a rare disease. And in Europe and in China, in Japan, the definition for rare disease sometimes it's not getting close, but slightly different. That's a part of the reason we don't see any particular regulatory guidance or guideline from the ICH. Even if we talk about the individual rare disease, back to 2 years ago, if we search the literature, and many of them, they were talking about 7,000 rare disease. Now, given the advancement of genomics, pharmacogenomics, so the number of the rare disease is increasing to over 10,000 different types. And collectively, in fact, it affects about just under 500 million people globally. And some of the rare disease is because of lack of awareness, and it's very difficult to diagnose, and take much longer time to get the accurate diagnosis. And the treatment for most of the rare disease, the treatment are not available. And if we're talking about the FDA-approved drug for rare disease, it only covers 5% of rare disease. And many of the approved drugs are only being used to treat one rare conditions. So here is some of the regulatory requirement, especially when I'm citing actually in my slide, I cited the requirement from FDA. And even the regulatory approval for common disease will require substantial evidence. Actually, the evidence standard, even for rare disease, not being considered differently from common disease. But it will touch a little bit, explain a little differently. For example, here I say, for example, in one of the FDA's guidance, substantial evidence of effectiveness of a human drug and biologics, it is said it can be approved through the accelerated approval pathways And the approval can be based on the one adequate and well-controlled drug plus confirmatory evidence. And this confirmatory evidence actually, in part of the confirmatory evidence, will include real-world evidence. Here, I will quickly go through the regulatory guidance. There's so many, as I said, there's no specific guidance or guideline from ICH, but the FDA, the US FDA has probably is the agency who has the most issued the most guidance or guideline for rare disease. And followed will be by the EMA. In my slide, I didn't present the regulatory guidance from EMA. I just present selectively one from the US and the other one from National Medical Product Administration of China. See, I don't read through the name of the guidance guideline. For those of you who are interested, you can get my slides, or you can go to the FDA website to search on the rare disease. Here's the FDA many guidance guideline from FDA. And this is a list of the guidance guidelines from National Medical Product Administration of China. Okay, challenge in rare disease drug development. As you could imagine, because the disease is very rare, and in the population size in general is very small, and because of the small population size, it will cause some of the problem to design a clinical trial, either the randomized control trial, or even for a single arm trial. Is sometimes you have a challenge to recruit a patient, and if the sample size is very small, and you can sometimes you only rely on the medical center, most likely located in a big city, and the evidence you generate from this kind of a clinical trial rely on the medical center will have generalizability problems, because many of the disease, they very geographically very spread out across all the country. Some of them live in remote village, and because of the disease is very real, and is a lack of accurate diagnosis, and the people are not familiar with the condition, clinical manifestation, etc., and don't pay attention about the disease, and the lack of specialized tests and procedures for the diagnosis, etc. Also, the natural course of the disease in general, the understanding of the natural history is very limited, and the genotyping and the genotype and the phenotype and the heterogeneity for the patient, etc. And the clinical, because we don't have much understanding about the disease itself. So in general, the lack of consensus of clinically meaningful endpoints to design a clinical trial, either randomized control trial or single-armed trials. So we need the clinical endpoint. Sometimes we use the surrogate endpoint. And this surrogate endpoint may not be necessarily translate into the clinical benefit of the patient. So that's another issue. For real disease, sometimes the progress was very fast, sometimes the progress was very slow. And in order to observe the clinical benefit of any treatment, sometimes we need to carry quite a long-term follow-up for the trials, and that will cause many of the people will cause the subject retention problems, another issues. And from trial conduct perspective, because if we consider the traditional clinical trial, we rely on academic medical centers. And that will have we only be able to recruit people who have access to medical centers, and for people who are geographically dispersed subjects not being able to be recruited to the trials. From patient perspective, lack of awareness and lack of trust. Why is the lack of trust? Because if you think about some of the people living in a geographically remote village, and the trust for these people who live in a remotely, and to the physician in the medical center, they don't have the mutual trust. And also for people who live in a remotely, they need support service, transportation, financial assistance, language translation, etc. And from community and healthcare system perspective, insufficient engagement for the healthcare professionals, and lack of a priority and prioritization and integration, etc. And some other the challenge from trial conduct perspective may include insufficient connection and partnership, inflexibility of the outcome assessment, and medical license. If you think about if the patient living in a borderline between the two states, and the physician can only have the license within one state, they don't have the licenses for the other state. That will cause a problem for the physician to treat the patient from other states, even the geographically, physically, the convenient location in the convenient locations. From data analysis perspective, that will also include part of being considered in the trial design. Say, for example, if we consider externally controlled clinical trials, and how can you choose the external control? That is the biggest issue. And even you were able to identify the external control and how the control is a historical control, contemporaneous control, or the mixed control, or the control data can come from electronic medical record or from a disease registry, etc. So there's a lot of other issues that we have to consider. Data analysis will have to consider heterogeneity in the patient population, and the surrogate endpoint, the clinical benefit, and statistical power, and this all the things that we have to make sure we have a maximum connect. And the surrogate endpoint should be able to carry some sort of like a practicability for the clinical benefit of the treatment. And benefit risk assessment is another issue. It's even more challenging even for the single arm trials. A single arm trial, you have the with external control, and because the patient population can come from very differently, and the data collection and the treatment option, and even the intercurrent event, for example, will be very different. So the benefit risk assessment will be even more challenge. And the strategy to deal with the challenge, and the previous we just discussed the challenge from statistical perspective. Now I'm going to briefly discuss the strategy to address this challenge. This is a diagram showing what kind of information we have. Based on the information we have, we can choose the development pathway for a rare disease. For example, if we have a clear understanding about the natural history of a disease, and we should directly go to the clinical trials. And if the natural history of the disease is very limited or partially known, and we should do the natural history studies. And how are we going to design for this based on what kind of parameters we're looking for the natural history study, etc. And for the clinical trial, basically two different types, with or without internal control. So without internal control is a single arm trial, with internal control, you can do the randomized control trials. Randomized control trial, it could be, and the patient can be unevenly allocated. Say if we have some of the or more evidence showing the treatment is more promising, and you can have a larger treatment group and smaller control group. In that case, you can still use external data to augment your internal control. That's the ideal situation, etc. So for those of you who are interested in this, you can download from Clinical Pharmacology and Therapeutics website. In our paper, we explain how to choose a development pathway for rare disease based on the information we have. Okay, study design. So adaptive design, of course, is very important one because the information at the beginning, we don't know much for the rare disease. And when we design a study, then we can use the biomarker, or the biomarker could be part of the development strategy for the trials. And we can use alternative designs, RCT with unequal allocation, a single arm trial, external control, master protocol, crossover design, randomized withdrawal design, etc. So, so when you have the information that is relatively uncertain, you can do adaptive design, enrichment design, etc. So depending on the information you have. And in general, for the clinical trial, we do the adult trial first, and then expand it to pediatric trials. And of course, the model-informed extrapolation can be used. And right now, it is one of the very popular areas. It's a model-informed trial design, and that's getting winning popularities. Especially in some of, say, for example, in dose finding, dose optimization from adult to pediatric population, extrapolation in these areas has particular applications. And use of the biomarkers. And the biomarker always remind me of so many different types of biomarkers. And here I just diagnose the biomarker, PD biomarker, or response biomarker, predictive biomarker, prognosis biomarker, safety risk biomarker, etc. So depending on where clinical question, we can choose the type of biomarkers. And most likely when you have in clinical trial, when you have infect modifier biomarker, we usually call the predictive biomarker. In that case, depending on the strength of evidence you have for the biomarker to modify the treatment effect, you can choose the different designs. Choose of the endpoint is another big areas, and we have the FDA, they have published rare disease endpoint advancement, a pilot program. And for those of you who are interested, you can look through. And sometimes for rare disease, especially when the disease is cells very heterogeneous in terms of a patient population, we can choose multiple endpoint to capture the treatment effect. I will not, the choice of an external endpoint, that's external control, that's another important area. I will not focus on that. Okay, use of, from study conduct perspective, a lot of publications describe how people use a real RWD to locate the disease, the patient with a rare disease. And we can use DHT, digital healthcare technology, to do the decentralized trials. And oh, by the way, there's another one, we have just decentralized clinical trial in the area of real world evidence. It's just we are very happy being promoted by the journal clinical and translational science, being promoted to as a position paper for that journal, just being published beginning of the years. SAT with external control, and we have a publication published in New England Journal of Medicine, New England Journal of Statistics and Data Science, telling people describing the scenario. The SAT will be the most appropriate for the sponsor to use in order to support the regulatory approvals. Multiple endpoint, I just discussed that a little bit more. Probability of the surrogate endpoint for clinical endpoint. I believe a couple of years ago, or at least probably the beginning of the last year, 2024, the ASA, Biopharma section, and FDA, they have a monthly meeting discuss the predictability of the surrogate endpoint for clinical benefit. And we have to make sure and the predictability is very strong. Otherwise, the use of a surrogate endpoint to support a clinical trial, especially for regular approval, will be very problematic. BIA, I just mentioned that, and okay, other consideration, the post-marketing study, expanded access program. For sometimes when you have a drug approval for a rare disease based on surrogate endpoint, and you can make the product to be accessible to after the accelerated approval, and then continue collect the data from an expanded access program to demonstrate clinical benefit, relatively long-term clinical benefit for people who use the product in the real world. Okay, let's move to the this one, the use of RWD and RWE to support rare disease drug development. And the first thing we have to consider for many of the rare disease, we don't have much knowledge about their natural cause of the disease, about the clinical manifestation, about the disease stage, about the biomarker, etc. A lot of things we don't know. In order to better design a study, we need to do natural history studies. And the purpose of the natural history study, say for example, to assist the trial design, determine some of the design parameters, the incidence rate, the prevalence rate for the rare disease, and precisely identify patient population, and with a certain, say for the biomarker status, the clinical stage, and the clinical manifestation, etc., and determine appropriate endpoints. And this is very important. We have to make sure we have a full understanding from natural history study before we design the clinical trials. And here, the FDA, they have a definition of what is the natural history studies. I will not read through and just mention that this is very important. And so when we design the natural history studies, there's a couple of things I want to bring up to your attention. No matter what kind of study we do, every scientific research they have has an estimate, either descriptive estimate or causal estimate. For the natural history study, most likely we have a descriptive estimate. So we have make sure and we have the definition. And if we look at the ICHE-UR1, one of the important components for the estimate intercurrent event. For the natural history study, in general, there's no intercurrent event, but we have to be careful about a competing event. Competing event, a special type of intercurrent event. And the other sensory event. Sensory event is a competing event is preventing the study endpoint from occurring. Why is the sensory event prevents the endpoint from being measured? So this is a little different, and we have to make sure. And when we have a precise and definition about the competing event and sensory event when we define the descriptive estimate in natural history studies. Inform the trial design, and we mentioned, say for example, we mentioned that before from a logistic perspective, and we can help site selection. We can look at the geographic distribution of the rare disease across the whole country. So for example, give you an example, follicular lymphoma, in the high prevalence can be seen for follicular lymphoma in New England area and in Southwest area, for example, Arizona, in New England areas. And in any other areas, and the prevalence rate is relatively low. If you want to enroll the patient faster, and you better to have more site open in New England and in Southwest areas, regions. So that's something we can study real world data to identify the patient geographic distributions. And from scientific perspective, and design the patient population, endpoint selection, biomarker, etc., and the sample size calculation, of course. And real world data, real world evidence that can generate the hypothesis about a certain certain things we want to know, especially about the surrogate endpoint level status or clinical status, the prevalence rate for certain clinical state of the disease, and given for the patient being receiving standard of controls. That will help us to design the study. For single-arm trial, we can choose the external control from electronic health medical record, or from insurance claim database, or even from the natural rare disease registry data. I don't know how many. What I heard last time, I heard there are about 130 different rare disease registries within the US. And you can imagine across the whole world, there are so many rare disease registries. That will help us to identify the patient, especially for the external control. The hybrid control of the RCT, this is very common. This is the one I liked it very much, because it has an advantage of to allow the sponsor to assess the similarity between external control and internal control, and also to augment your study power with external control. So it has the beauty of this for the hybrid control for RCT. And patient engagement and the trial promotions, you can have the target engagement and promotion during the trial. And trial planning, I mentioned that before from trial conduct perspective. And patient identification, we have been, actually our company have been using the artificial intelligence machine learning algorithm, and to match up the patient in our database or in individual medical center database or any other specific database being registered in that database, and to identify eligible patient, preliminarily eligible patient for rare disease trials. And descriptive analysis, that's the third piece of use of RWD and RWE. The first one I'm talking about the study design. The second I'm talking about study conduct. And the third piece will be to support data analysis and descriptive analysis as a natural history study. And most likely we use the disease registry or electronic medical record. These are two real RWE data type, data sources are most suitable for natural history study. Of course, you can do either longitudinally or cross-sectional. Each of one, they have unique advantage or disadvantage to conduct a natural history study. Comparative analysis, there's what I said, the rich literature on this regard. So many publications talking about comparative effectiveness, either using single-arm trial with external control, or simply non-interventional observational study data, either existing prospective or retrospective data. Okay, what else I have? Data analysis. Okay, I have 22 minutes. I will make sure I leave at least 10 seconds for Q&A. Analytical method, in terms of analytical method with using external data. This is the one I consider will be very good one. Say RCT with external control to augment internal control. You can do the test and then pull method. Using the Bayesian hierarchical model or Bayesian borrowings, regression adjustment, which is simply the G computing method for the confounding adjustment. Matching stratification with the propensity score, or now is getting people gradually picking up. In addition to propensity score, you can also use a prognosis score and risk score for the matching and stratification. Even for the probability weighting, Taimei is the one, and I really liked it a lot. And it has some of the unique advantage on this. And now we have given the increasing popularity for the AI and the machine learning, and we use the data adaptive approach for the causal inference, and actually quite often right now. Now I spent about 3 minutes talking about the ultra rare disease. Ultra rare disease, and rare disease is very rare, but ultra rare disease even more rare actually. And if we're talking about 10,000 rare disease, different rare disease type, 80% of them are ultra rare, and even some of them we'll be talking about globally. Only 20 or 30 prevalent cases for each of the ultra rare disease. So you can imagine from a treatment perspective, and it's very immensely more demanding, and most of the ultra rare disease are undiagnosed or misdiagnosed because we don't know, we don't have understanding about the disease itself, and increasing genetic complexity. And the other one is the lack of biological samples. We don't have biological sample, the patient data, scarcity of the endpoint, heterogeneity of manifestation, clinical manifestation, etc. So you can imagine, given all of this challenge, the use of RWD and RWE to support ultra rare disease is much needed. And again, we use a disease registry and even EMR, EHR, and plus and AI, the machine learning, etc., to identify this patient from this databases. Here I gave the example for those of you who are interested in, they can look through. And cell and gene therapy is getting popular in ultra rare disease areas. Target learning, I really like it a lot. We have the whole RWE scientific working group highly recommend this in our evidence generation from either controlled clinical trials or real-world evidence studies. Why we said the controlled clinical trial, we still prefer the target learning, because if you have a randomization, at the point of the randomization and the baseline characteristics of the patient are approximately balanced. But after that time point, and the baseline the patient characteristics start losing balance, because the patient either switch treatment or drop out for whatever reasons, and not continuously taking the original assigned treatment. So that's a causal problem we have to deal with, the dropout, the treatment switch, or any other intercurrent event. So okay, here is the target learning based framework. Sometimes you see 7 different steps or 6 different steps. You start with the research question you're trying to answer, and given that research question that you precisely define your research, your study estimate. And now then you have observed data. This observed data distribution either come from existing data, say for example, if you choose external control, historical control, and the data already there, you can choose, and you have the observed data. You can describe using the data generation mechanism to describe the data, the distribution, and then define statistical model and target statistical estimate. Either, for example, you can do multiple regression adjustment to estimate the causal effect, or you can do the hierarchical modeling, and then you can define your statistical estimate. And then use your causal knowledge, use your domain knowledge to define the causal model and the causal estimate. And then you have a statistical estimate, you have a causal estimate. And the difference between the two will be your causal gaps. This causal gap will be something you worry about. Why? If it's very small and the statistical estimate and causal estimate very close, now the cost gap is very small, which means the assumption, whatever the assumption you have, either statistical assumption, missing data assumption, and causal assumption, the impact of these assumptions on the causal gap is minimal. But if the gap is very large, and then you have to thoroughly study, do the sensitivity analysis to see what kind of causal assumption will most likely change your conclusions. Again, the definition of the estimate is an iterative process. Sometimes you may choose the data, and sometimes you find out, and the data you have will not be able to answer your originally posed questions defined in your study protocol. Now you have 2 choices. One is to refine your original research question, or to find another data set. If you don't have another data set, and then you have to refine your research question and the corresponding estimate. Okay, I have quickly go through, I believe, 2 or 3 case studies. The first one is about natural history studies. And this MCC is a very real aggressive skin cancers, and there was no innovative therapies until the FDA approved the drug, what's the anti-PD-1 drug back to 2017. And the study was called Javelin Mercure 200 trial. It's a single arm of trials, and use the overall objective response as a primary endpoint and DOR as a key secondary endpoint. And 88 patients being enrolled with a minimum 12 months follow-up. Individual review committee, the overall objective response rate was 33%. And here's the 95% confidence interval. The median DOR was not reached. So this is the trial data from the single arm trials. And a retrospective charter review in US Oncology Network identified total 39 patients with metastatic MCC, and 14 patients with a similar eligibility criteria for the patient in these trials. The OR was 29%, and median DOR was 1.7 months. You can imagine, here's the 29 from the real world. This is a natural history study. And 33 from single arm trial. 33 is only about 5% higher for the ORR. But here is the DOR is only 1.7 months. Here's a make a big deal. 5.7 months. It's a minimum 12 months follow-up. The median DOR was not reached. I guess this make a big difference. The other one, the pivotal immunosuppressant for organ transplant. Okay, let me see. This is used U.S. Scientific Registry transplant recipient data for the primary lung transplant between 1999 and 2017, with a composite endpoint of a time to graft failure or death. Here is the trial data. Okay, let me see. Yeah, okay. Here is identified adult patients is a 15,000 over 15,000 of 450 pediatric patients with a 1 year graft survival time of the 91, nearly 91%, nearly 92%, 91% for adult and 92% for pediatric. The Prograph MMF concomitant medication. The other one, the Prograph from the AZA with a 1 year graft survival estimate is also 91%, 91% for adult and 85% for pediatrics. And it was generally believed this result was deemed very unlikely to have occurred by chance alone compared to the document the natural history of the transplant lung with no or minimum immunosuppressant. So this is use real world data to generate the treatment effect for the combination of Prograph with one of them, and is generally believed that this generate a very good result. And what we said, a pivotal role for RWD and RWE for regulatory decision. The third one for, I guess probably you're very familiar with this, about the Pfizer's breast cancer for men, the drug. And it didn't, the study, let me see, okay. Here is the approval history for Ibrance, the grant, the first in 2015 for women, and 2016 for HR1 plus HER2 and metastatic breast cancer. 2017 for first line HR positive and HER2 negative metastatic breast cancer. Supplemental NDA in 2019, the treatment for men with HR plus and HER negative metastatic breast cancers. So this approval actually is based on the RWD study from EHR and the post-marketing report of the real-world use of Ibrance in male patient source from 3 different databases. Okay, let's see how this study will generate 2 cohort actually. The 2 cohort A for the Ibrance treated cohort, and the cohort B will be for the patient non-Ibrance treated cohort. And you see the cohort A will give the observed response rate 25% in cohort A. And for cohort B, the only less than 13% for patients not being treated with by Ibrance. And considering the mechanism of the drug and also extremely rare for the breast cancer in men, so FDA approved the label expansion for this drug. Okay, some of the conclusions, and here I didn't talking about very much, just briefly mention when you do the external use external RWD, RWE, and first thing you have to think of whether RWD is fit for purpose. Here are the 3 dimensions of the highly recommend and being thoroughly discussed in this paper by Mark Levinson and his colleague. Relevancy and reliability being discussed by the FDA in the framework. Fit for research consideration is the data prevalence, traceability, and the data standard. That's very important. That will consider being part of data quality, either especially when you have a regulatory decision perspective. You have to make sure that data traceability is very important. And construction of an estimate involving external data. The complexity is very high, and especially the intercurrent event. You have internal data and external data, and the patient may come from different population, and the pattern, the frequency and the pattern in the current event may be substantially different. In this case, what kind of a strategy I can consider to deal with in the current event? And I said here is I have mentioned every treatment effect among treated, every treatment effect among untreated, and sometimes every treatment effect and overlapping population of ATT, ATU, ATO, they refer to different things. Their assumption, definition and assumption will be very different. For this type of estimate, you need to consider the complexity to have to provide a thorough scientific analysis to explore the impact of the assumptions on your study conclusions. Scientific analysis, this is something being discussed recently, and depend on the potential, the occurrence of a potential bias. And we usually perform the scientific analysis in a reverse order of the bias occurrence. Here is the assumption, say for example, confounding bias usually in the source of a population often occurs before selection bias, which can take place before information bias and distributional bias. And in this case, we probably want to do scientific analysis to first explore the distributional bias of assumption first, and then information assumption. After that, we do exchangeability assumption and non-major confounding assumption, etc., in the order of to the scientific analysis. Some of the take-home message is a wide range of application of RWE in clinical trial or clinical development for medical product. Some of the special attention actually are required for their use, especially in ultra-rare disease. And we have to consider in many different ways in the study design, conduct, patient selection, site selection, data analysis, bias minimization, scientific analysis, etc. Again, target learning based framework is highly recommended to provide a systematic approach for causal effect estimation in a transparent and interpretable manner, and which it's very important, I believe, in the RWE generation for rare disease drug development. Okay, I have a whole bunch of the references I listed here at the end of my slide. For those of you who can send the email to the host, request my slides. And thank you so much. That's the end of my presentation. Open for any questions you may have.
Thank you very much, Dr. Chen, for the very comprehensive introduction and the insightful your ideas. So I have a question. So given the challenges of mirrored confounding and non-exchangeability, if we're using external control study, the causal gaps you mentioned, right, become a very critical concern in regulatory decision making. So I'm just wondering, are there any regulatory bodies such as the FDA, EMA, that have explicitly accepted some specific statistical techniques or sensitivity analysis to address these kind of gaps if in the real world evidence studies.
Yeah, very good question. I remember back to about 2 months ago when I received for one of our manuscript, we received a comment from a referee. One of the referee asked the same questions. We actually, in our manuscript about the RWE, use of RWD and RWE in rare disease drug development, and for each of the steps for the target learning framework, we provide a running example how each step will generate what, and will be how can we interpret the result, and what will be the regulatory acceptability for each of this. And for those of you who are interested, actually, maybe I can send you, I'm not sure if it's public available, it's downloadable for the PDF. I can send you the example. Did I answer your question?
Yes.
Okay, great, thanks. Actually, I'm still waiting for, on the FDA website, it specifically mentioned the demonstration project for the target learning framework. I would like to definitely be interested in the assessment report. Unfortunately, I don't have.
Yes, sure. So, are there any questions from our audience? You can unmute to ask one question. I also didn't see the chat in the question in the chat. Okay, so I think if no questions, I think that it's also a good timing. And I want to thank you again, Dr. Chen, for your insightful presentation and for sharing your expertise with us today. And also big thank you as well to all our attendees for your participation. And we hope you found this session valuable. As I just chat in the chat box, the recording and the slides will be shared later. And we appreciate your time and look forward to seeing at our future ISBS webinars. Have a great day. Thank you very much.
Thank you. Thanks, Taidao. Bye-bye.
Bye.
Bye.